very much again, Dr. Farsh, for inviting me to participate at MedTex Day. Today, I am going to talk about my project, which is called Anti-fibrotic Effects of the Human Adipose-derived Mesenchymal Stromal Cells Secretome in Vitro Assessment. So first of all, why are we interested in fibrosis? Well, fibrosis is a pathological scarring process which involves at least 4 of the 10 leading causes of death globally. It's also an irreversible and generally progressive process, which is involved in at least 50% of mortality in developed countries, and is affecting not only organ-specific disease like heart failure, chronic kidney disease, cirrhosis, interstitial lung disease, but also generates systemic disease like less prevalent, but definitely prototypic systemic disease, which is systemic sclerosis. So to understand what is the difference between the physiological process of wound healing and fibrosis, I will show these images that explain the 4 main steps of wound healing and how they differentiate from fibrosis. So when an injury occurs, an immediate trigger generates a clot to stop the bleeding. Afterward, inflammation with limited the microgram invasion and limited the debris will activate inflammatory cells and also the migration of fibroblasts, which will be activated and will proliferate and synthesize extracellular matrix components, which will compose the granulation tissue. These finally will be no longer needed, so myofibroblasts will be induced into apoptosis, and the degradation of extracellular matrix will be generated, allowing functional cells to regenerate. Now, what happens in fibrosis? Inflammation and proliferation persists, generating the myofibroblast survival and producing excessive extracellular matrix components. So fibrosis is a pathological scarring process that is characterized by the excessive production of extracellular matrix components, mainly collagen. So which is the main cellular effector of fibrosis? This is myofibroblast, derived mainly from fibroblasts, also from other cells. And among the pro-fibrotic cytokines and growth factors, TGF-beta is one of the most important growth factors that promote myofibroblast survival. Myofibroblasts are defined as cells that are capable of contracting due to stress fibers that are composed of alpha smooth muscle actin, and also are capable of synthesizing extracellular matrix, mainly collagen. This is why you'll see in my following slides that we use alpha-smooth collagen as our reader. So here we can see the canonical pathway that is triggered after TGF-beta binds its receptor, and as a result leads to the transcription of the target genes that promotes fibrosis and myofibroblast survival. There are different antifibrotics that are being used in the clinical practice that attempt to inhibit this cascade, but none of these can revert fibrosis so far. And here is when we are so interested in cellular therapies as possible antifibrotic therapies. So mesenchymal stromal cells were initially characterized from the bone marrow. Now we know that they can be obtained from different tissues like adipose tissue, umbilical cord, and these are defined by the International Society of Cellular Therapy as plastic adherent cells with fibroblast shape that are positive for CD90, CD73, CD105, and negative for hematopoietic progenitors and negative for HLA-DR. Also are capable of differentiating in vitro into adipocytes, chondrocytes, and osteoblasts. The main mechanism of action is due to the paracrine effects by secreting soluble factors and extracellular vesicles. And so far, there are in vitro assays to test the immunomodulatory capacity of mesenchymal stromal cells. Also, we have developed in vitro assays to establish the pro-angiogenic effects of MSCs, but there are yet no standardized in vitro assays to establish the anti-fibrotic effects of MSCs. So our hypothesis is that the MSC secretome inhibits pro-fibrotic mechanisms, and our aim is to establish in vitro assays to assess the anti-fibrotic effects of conditioned media from adipose tissue-derived MSCs. In order to do this, we isolate MSCs from adipose tissue. We culture them under resting and inflammatory conditions and collect the conditioned media, which was tested on immortalized human foreskin fibroblasts. So the first in vitro assay, we call it inhibition of fibroblast migration. Here we establish vitro two different densities of fibroblasts, and we did a linear scratch. Afterwards, this was treated with conditioned media from resting and primed MSCs, and images were taken every 3 hours for 72 hours. The cell density was analyzed by a scrunch-wood cell migration software. And as we can see here, this is an example of the images obtained at the initial time point and after 48 hours. And there is a decrease in cellular density in the fibroblasts which were treated with conditioned media from MSCs. In these graphs, we can see the results that we obtained in blue, the controls, and in red and green, the samples that were treated with conditioned media from either resting or primed MSCs. And as we can see, there is a decrease in the cellular density compared to the controls, and there was no difference between the conditioned media that was obtained from resting or primed MSCs. Here we are showing the summarized data that again shows how there is a decrease in the wound density compared to the controls. The second in vitro assay is called prevention of fibroblast differentiation. The rationale here was to test if conditioned media from MSCs can prevent the differentiation from fibroblasts into myofibroblasts due to TGF-beta. So we simultaneously treated fibroblasts with TGF-beta and conditioned media from MSCs, and after 72 hours, we collect the proteins. Here I'm showing images as an example of the Western blot that were obtained. We can see fibroblasts treated just with TGF-beta, and those fibroblasts which were treated with TGF-beta and conditioned media from resting MSCs and primed MSCs. And in the graph, I'm summarizing the data, again showing that there was no increase in alpha-SMA and procollagen in the samples that were treated with conditioned media from either resting or primed MSCs. In the third in vitro assay, which we called myofibroblasts into fibroblast dedifferentiation, which is one of the most interesting for me, we first differentiate fibroblasts into myofibroblasts, and after 72 hours of treatment with TGF-beta, we add conditioned media from MSCs, and after 72 hours of treatment, we collect the proteins. Here I'm showing again the images that we obtained from the Western blot. And here we can see a reduction in both alpha-SMA and procollagen when the fibroblasts were treated with conditioned media from either resting or primed MSCs. As I was saying, this for me was one of the most important in vitro assays because these show the differentiation from myofibroblasts into fibroblasts when the fibroblasts are already activated by TGF-beta. The following question here was, for us, was if conditioned media was killing myofibroblasts or de-differentiating them. So by flow cytometry, we measure cell survival. And as we can see here, after 72 hours of treatment, we see that the percentage of cell survival is the same. So as a conclusion, we can say that so far, in vitro conditioned media from MSCs can inhibit fibroblast migration, can limit the differentiation from fibroblast into myofibroblast, and promotes the differentiation from myofibroblast into fibroblast. The implications of these in vitro assays to the in vivo anti-fibrotic properties still remain to be defined. So finally, I would like to thank my supervisors for their continuous support and teaching, and also Maximilian Laura, who is the research assistant that has been teaching me so much, and all the team that is helping me to develop this project.